Hi, I'm Raizel, and we're going to be talking about phytoplankton in the Gulf of Maine, and we were trying to figure out, is nitrogen a limiting nutrient?
I'm Madi.
I'm May.
And I'm Megan.
We have some examples here of phytoplankton. At the top right is Navicula. Down at the bottom we have Thalassiocyra. We also have Alexandrium fundiense on the left, on the bottom. And we have Leptis danicus on the top left. These organisms produce 50% of the world's oxygen.
This means that every other breath you take is thanks to phytoplankton.
Without them, the world would not be the same, and every ecosystem would be affected.
But even though we need phytoplankton, they can also be deadly not just to humans, but to our oceans as well. Phytoplankton are very important because they are the ocean's primary producers, meaning that they serve as the very bottom of the food chain. They can also be key indicators of climate change, as their reproduction directly depends on the water temperature and the amount of nutrients. This can be indirectly controlled by the winter-spring bloom, which is a phenomenon that occurs in the changing of seasons between winter and spring, where there's more sun and more nutrients, which are both key factors in phytoplankton reproduction. And this is a natural process that occurs annually.
Nitrogen is a key nutrient for phytoplankton, which are microscopic photosynthetic organisms. Here shows the beneficial and harmful effects of the presence of nitrogen. The winter-spring bloom is an annual natural occurrence, especially along the coast of Maine, and hypoxia and harmful algal blooms like the red tide from Alexandrium fundiense are some harmful effects when nitrogen is introduced in excess.
So Madi just mentioned something called hypoxia, and this graphic basically shows what that is. So what happens is excess nutrients like nitrogen and phosphorus are entered into the ocean, usually through runoff from fields of fertilizer or sewage, and these excess nutrients cause phytoplankton to bloom. This is not a natural process because the nutrients come from human pollution, and the excess phytoplankton aren't able to be eaten by organisms in the upper trophic levels like fish and mollusks. And so some of the phytoplankton die and they sink to the bottom of the ocean where their biomass is decomposed by bacteria. But the problem is that these bacteria use oxygen and they produce carbon dioxide. So what happens is we have these dead zones in the ocean where there's very little oxygen and a lot of carbon because bacteria have used all of the oxygen.
So we just looked at hypoxia, which was one of the harmful effects of nitrogen, like nitrogen pollution, which actually comes from things like fertilizers and sewage. Those are the main culprits. And now we're going to take a look at harmful algal blooms, like the red tides I described earlier. So Alexandrium fundiense is a toxic dinoflagellate that can appear in the Gulf of Maine. It is photosynthetic because it is phytoplankton, and it causes red tides and it produces toxins that lead to paralytic shellfish poisoning, which actually kills humans if we ingest too much of that toxin. And one commonly referred to red tide is occurred in 2005, and it caused Massachusetts to go into a state of emergency, and it also caused the shellfishing industry to close down, and it cost them $50 million. So it's a huge problem.
So after looking at all these different processes, we came up with 3 hypotheses. And our first hypothesis, the one that we really focused on, was that when we increase the abundance of nitrogen, we would see an increase in the abundance of phytoplankton. But we also hypothesized that not every species would respond in the same way to that increased nitrogen. And we also wanted to look specifically at Alexandrium fundiense, and we hypothesized that we would find more Alexandrium in shallower, warmer, lower salinity waters close to the coast where there's lots of runoff containing nutrients.
So, in order to collect our data, we went to several locations, including Maine Maritime Academy. At Maine Maritime Academy, our group went out on the RV Friendship to collect water samples from both up bay and down bay. Up bay means closer to the shore, while down bay was taken in the open ocean. We actually took the up bay directly from the docks. We used their CTD technology, which is an oceanography instrument used to collect water and determine its conductivity, which means salinity, temperature, and depth, to take samples from the two locations. We sampled water from both up bay and down bay in order to study the differences in types of species and the abundance of each location. We also were curious to see if one location responded differently to the added nitrate than the other. We then took a sample from Casco Bay. This location is an estuary, and the salinity of the water we took was only 5 parts per million, compared to 30 parts per million in the Penobscot Bay. It is much closer to the coast than either of the Penobscot Bay samples. Additionally, where we collected it from Casco Bay, the samples were right down the road from the farm, which means that there was probably a lot of runoff, which would affect the samples. We concentrated the water from each location using a plankton concentrator, a PVC pipe with a 20 micron net, to concentrate the plankton so that we didn't have to filter through as much water. We fixed these samples so they didn't decompose. We took the seawater from each location and incubated it with nitrate and kept some controls with nitrate. We incubated the samples from Penobscot for 7 days and the Casco for 4 days. We counted the fixed samples under a dissecting microscope and counted the abundance of the cells to identify which species of phytoplankton were present. We then used the fixed samples and used them, they act as a control so we could observe exactly what the plankton populations looked like when we first captured them. We ran the whole water sample through a flow cam, which took pictures of the samples and explained which species we had. So this is a picture of us using the flow cam. The flow cam, you can look at each species, all species you have, and then click a singular species and identify all of them within the sample in order to calculate abundance. We used the software called Visual Spreadsheets, which sorted the pictures into each species, and then we calculated the abundance per millimeter.
So after analyzing our data using the FlowCam, we saw that there was an increase in the abundance of plankton in the samples from each location. So if you look at these graphs, the x-axis shows whether the sample contained, was the control or the sample with added nitrate, and also which location we took the sample from. And then the y-axis is the cells per milliliter. So you can see that we saw a pretty dramatic increase in the abundance of cells between the control and the nitrate at all 3 of our sampling locations. But when we broke down our results by species, we saw that not every species responded the same way to the increased nitrate. So you can see up there we have a graph showing Crassostrea, which are these cylindrical chains of diatoms. And you can see that Crassostrea showed an overall increase between the control and the nitrate at both the up bay Penobscot location and the down bay location. But the down bay sample had a much more dramatic increase than the up bay. Whereas if we look at Thalassiosira up there, it's also a cylindrical chain of diatoms. You can see that the increase was much more proportional between the up bay and the down bay samples. But when we look at Navicula down there, you can see that we actually saw a decrease in the number of cells between the up bay sample and the down bay sample. So this probably means that Navicula don't need nitrogen as much as other phytoplankton do.
In addition, we looked at the abundance of oxyspores, which are these little guys. And you can see that they're different from other cells because they have this little end coming off, and that's because oxyspores are reproducing cells. And this is the top picture is a picture from our data that we found. And the presence of these oxyspores would suggest that the phytoplankton are thriving in their environment. And this pie chart depicts the abundance of oxyspores in each sample. And it shows that the oxyspores were only present in the up bay control and nitrate and only the down bay nitrate. And they had a much higher presence in the 2 samples with nitrate here because it would show that the phytoplankton survived better in the nitrate samples and were able to have the resources to reproduce as well.
Going back to Alexandrium fundiense, we looked in the fixed samples to determine whether or not our hypothesis was supported, and in general we found that it was, since 84% of the Alexandrium fundiense that we found in general was from Casco Bay. And we depicted that was true because Casco Bay is shallower, it's more southern than Penobscot Bay, it's warmer, and it has a lower salinity. And also it's closer to the farm, so it still has that nitrogen that could be coming from the farm. And we also observed that we found more Alexandrium in the up bay sample than in the down bay sample, which we also thought had to do with the presence of nitrogen.
So to recap our results, we found that all three of our hypotheses were supported. When we incubated our seawater samples with nitrate, we found that we saw an increase in the abundance of phytoplankton, but we also saw that not every species responded in the same way to the increased nitrate. And we also found that Alexandrium fundiense was the most abundant in our sample from Casco Bay.
So on the bottom here, you can see two headlines that are about red tides that have happened in the past. And this is important because the number of harmful algae blooms are increasing in the Gulf of Maine due to more nutrients. And because of the geography of the Gulf, it's harder for those nutrients to escape to the open ocean because there are all these islands that prevent it from getting out. And our study proves that adding nutrients to the environment has a dramatic effect on the abundance of the phytoplankton. And in addition to the red tides, Alexandrium actually aren't the only type of harmful algae, because there are other harmful, non-toxic algae that can become just as harmful when there are excess nutrients.
So what can you do to help decrease the number of harmful algae blooms? One important thing is to limit the increase of nitrogen in our marine ecosystems. So a really easy way to limit the output of nitrogen is to stay away from commercial fertilizers, because they're designed to have all these added nutrients that when it rains and they're washed into the ocean have a really dramatic effect on populations of phytoplankton. And another thing to do is to plant cover crops to reduce runoff and to help absorb these excess nutrients, and also to support agricultural businesses that have clean runoff and that don't use these commercial fertilizers, as well as to fight for legislation that will help regulate nutrient pollution from agriculture.
We'd like to thank Dr. Carrie Whitaker, Claudia Mazur, Laura Carson, and everyone at the Flow Camp who helped us, as well as everyone at Maine Maritime Academy. We couldn't have done it without all of you. Thank you to our students who supported us and who went out to do collections with us. It's been really fun. Thank you.